Welcome everyone. This is the first lecture of Bio 139 lecture. I am your instructor, Scott Davis. I may have had you previously in Bio 137, or you may have had another instructor but still had some experience with me with my Bio 137 YouTube lab videos. I may have had you previously for Bio 139 and you're retaking with me, or you may be brand new, but regardless, I want to welcome everyone to the class and hope to make you feel comfortable. This is, like I said, the first lecture, and we're going to do a little bit of an introduction in this video, and then we're going to dive in and actually start covering some of Chapter 18, Blood. There will be a few videos over this chapter, and I'm going to number them in order so you know which order to watch them in. Today's agenda, there is actually a separate video that you should have watched before this, posted to Blackboard, in which I cover the syllabus for both lecture and lab, the Blackboard structure, where to find everything, course layout, what to expect in this class, how to do well is posted in Blackboard, but kind of building on that, before we get rolling, I want to talk about how you should actually approach the lecture portion of this class. I'm about to start lecturing over the first portion of Chapter 18, Blood, but you should have already read that chapter. If you have not already read Blood and filled out the course outline for Chapter 18, Blood, go ahead and pause this video, stop it, come back later, because you're really going to get a lot more out of it if you read the chapter first, fill out your outline first, and then watch this video. The reason that is because when you read the chapter the first time, it's really not going to make sense, but you're getting exposure to the material. And if you fill out the outline while you read, you're kind of solidifying that material. But when you're watching the video, you're not trying to listen to everything I say and see what's shown on your screen and fill out the outline, all while I'm talking about stuff that you've never even been exposed to before. But by reading the outline, by reading the chapter and by filling out the outline first, that gives your brain more room to actually pay attention to what the lecture is talking about, and then it gives you a little bit of time to add extra notes to your outline that maybe you didn't get the first time through. Once you're finished watching the video and you've finished filling out your outline, go back and reread the chapter. And now not only have you read it once already and taken notes, and now you've listened to the lecture, when this second read through of the chapter happens, it's going to make a lot more sense. And I know in online courses that can be kind of tough to discipline yourself to do that, but it's really, really going to help if you do it that way. So, before we begin, now in an in-person class, this is when I pause to ask everyone what kind of questions they have before I ever get rolling. Now, with this being online, I can't really look out into the room and see who has their hands raised, but what I do is encourage you to text me or email me those questions. Don't wait to ask questions later on. Ask them when they come up. I'm always very, very responsive. As long as I'm awake and not busy eating or showering or something like that, if you text me, I will text you back within just a matter of a few minutes, maybe an hour at most. If you email me, I still get back to you quickly, but my phone doesn't always notify me, so sometimes it takes me a little longer. But please ask those questions. It can be kind of tough in an online class to think about even asking questions, but I'm still responsive just as I would be in an in-person class. All right, so think about what questions you have, and if you have them already, pause the video, text me, email me, let me know what those questions are before you even go any further. So, like I said, the first chapter we're going to talk about is blood. And blood at first can seem kind of boring, kind of like a small topic. But what I like to tell my students is blood is actually the central topic for this entire course of Bio 139. Remember back in 137, it seemed so just kind of thrown together. At first, you're learning the basics like the body cavities or anatomical position, and then you're talking about chemistry, and then you're talking about the cell, and then you're talking, one week you're talking about the skin, and then all of a sudden you're talking about muscle or bone or nerves, and it didn't really gel all that well. But in Bio 139, everything is going to come down to blood. To begin with, we're going to talk about, what is blood made of? What does blood do? How does blood work? And there's a lot of work that blood does. Then we're going to talk next about the cardiovascular system with the heart and blood vessels. So we're going to see, how does the blood get moved through the body? After that, we're going to talk about the respiratory system and see the reason that we breathe is actually to regulate the pH of our blood and the carbon dioxide levels in our blood. Next, after that, we're going to talk about the immune system and the lymphatic system and see, how does our blood work to keep us healthy? After that, we talk about the kidneys and the urinary system and see, how do our kidneys work to keep our blood clean? And again, like the respiratory system, the kidneys will also regulate our pH. After that, we're going to talk about pH in a deeper topic, things that can go wrong with your body's pH, and how do things regulate our blood pH. And then we're going to wrap it up at the end with the endocrine system. And the endocrine system is going to use hormones to control different parts of our body. How do those hormones get from one place to another? With the blood. So everything in this chapter, everything in this entire course, Bio 139, is going to come down to blood. So this chapter, even though it may seem kind of irrelevant, it is actually the most relevant thing that we cover this semester. So let's begin with an overview of blood, and then we're going to talk about plasma, and that'll wrap us up for this lecture. So, overview of blood. What are the functions of blood? There's several. Well, transport, like I just mentioned, things move around in the blood. So, blood takes stuff to and from various parts of our body. For example, it takes oxygen and nutrients to our tissues. It takes waste to our kidneys. It takes carbon dioxide to our lungs so that we can exhale it. It also transports stuff away from certain parts of our body. When we eat, it takes nutrients away from our intestines so that it can deliver them to the body. It takes oxygen away from our lungs. It takes waste and carbon dioxide away from our tissues. It also transports endocrine hormones. Like we just said on the previous slide, when our endocrine system releases hormones, it's blood that actually transports those hormones from the gland that released them to the part of the body where they have their function. Blood functions for protection. Later in the semester, when we talk about the immune system and the lymphatic system, we're going to see various cells and proteins in our blood that help fight off infections. Blood also protects us from losing blood. Blood protects itself from being lost. Later in this chapter, we're going to see, when we get an injury and we start to bleed, how does blood actually stop that bleeding? Blood has various regulatory functions. It regulates fluid distribution. When we have a drink of water, how does that water get moved from our intestines to the parts of the body that need it? When we're starting to get a little dehydrated, our blood actually moves the liquid in our body, the water, from place to make sure that it's evenly distributed. pH maintenance. Several chapters this semester we're going to talk about the pH of blood and how that pH is maintained. And you'll see I have some pet peeves about pH because pH balance is a term that everyone has heard, but it's a marketing gimmick. If you ever hear pH balance, just close your ears. And unfortunately, our book actually uses that phrase a few times, but it doesn't mean anything. pH is a measurement, just like inches. You don't talk about inch balance or pounds balance. pH balance doesn't mean anything. pH is just a measurement. Thermoregulation. This is how our body maintains a certain temperature. When we're too hot, we start to sweat, and the blood actually moves closer to the surface of our skin so that as that sweat evaporates, it cools our blood, and then that cool blood circulates through our body to cool down our organs. When we get too cold, we start to shiver, and blood moves away from the surface of our skin so that it's not closer to that cold air. The composition of blood, and what we see here in this image is actually a microscopic view of blood, just to kind of give everyone an idea of what you're looking at. These things that look like little pink Cheerios or donuts, those are called red blood cells. We'll see another name for them later. These big, weird-looking things with the purple blobs, those are white blood cells. There's a bunch of different types. And then we have these small little flecks that almost look like dust on the slide. Those are platelets. And then there's all of this white background, which we'll talk about here in just a moment. So blood is an unusual tissue, and it is a tissue. A lot of people don't think of blood as a tissue because it's a liquid, but it is a tissue. It's actually a connective tissue. Why do we know it's a connective tissue? Think back to chapter 4 or 5, depending on when you took Bio 137, when you were learning tissues, and you talked about connective tissues. The reason something is a connective tissue is because of where it came from when we were just embryos. When you're an embryo, there's 3 original layers of tissue: the endoderm, the ectoderm, and the mesoderm. And the mesoderm produces all connective tissues. Blood came from that mesoderm, sometimes called the mesenchyme. So that's why blood is a connective tissue. Another thing about connective tissue is the cells are not tightly packed. There's a lot of space in between them, and that empty space in between the cells is called the matrix. Now, if we look back at our picture here, we can see that there's a lot of space, this empty white space in between the cells. So that's the matrix. But in blood, we actually call the matrix something else. We give it a special name. This is blood plasma. So the plasma of blood is the matrix of the connective tissue. So we can actually separate blood using a centrifuge to see the different components. And that's what we see right here. They will draw blood, they'll put it in this little thing called a centrifuge, and it spins the blood really, really fast. And depending on how dense something is, it will actually settle further or not quite as far. And that's what we see here. The densest stuff sinks to the bottom. And this is actually where we find the red blood cells, which are actually scientifically called erythrocytes. We'll get to that word a little later. But the red blood cells down here at the bottom. Then we have the plasma, which is the least dense. Plasma is the liquid part of blood, and it's up here at the top. But there's a really thin layer right in the middle. This is where we find the white blood cells and the platelets. This is called the buffy coat. So, 3 things are separated out when we put blood in a centrifuge, and that's what we see over here. Down here are the red blood cells. Here's that buffy coat containing white blood cells and platelets. And here's the plasma, the liquid part of blood. Now, I want to introduce another term that we'll talk about a little later on, and that's the hematocrit. So when we look at this blood and it's been separated, we said down here at the bottom is the erythrocytes. And erythrocytes in the average, healthy, well-hydrated adult male, erythrocytes make up about 45% of your blood, just under half, and we can see that this is just under half. Well, the amount of red blood cells, this 45% or whatever it happens to be in our sample, that is called the hematocrit. Hematocrit is the percent of whole blood made of erythrocytes or red blood cells. Just as buffy coat is the portion of blood made of leukocytes, white blood cells, and platelets. So now I've given you some percentages there, and throughout this entire course, we're going to talk about percentages or weights or different values of things. And this chart that you see here gives some of those values for blood. This chart is in your textbook on page 672. And when we see values, sometimes it shows us both for a female and for a male. Sometimes our book doesn't do that. Sometimes I will just say the weight is, and I'll give you a weight or something like that. If it doesn't show it for both female and male, the number applies to the average healthy adult male. That's what I have over here, AHAM. The average healthy adult male. And that's just because average, we're not talking about someone who is somehow different from average. We're talking about healthy because we're not concerned with the sick person, we're comparing them to the healthy person. Adult, because when you're a child, these numbers are vastly different. And male, just because they have to pick one, and for whatever reason, male seems to be the standard. All right, so here are the general properties of blood using that average healthy adult male in most cases. But again, here we see female, male. Down here we see female, male. Now, you do not have to know everything on here. Now, by the time we get to the end of this course, you're going to know a lot of this stuff just because we talk about it a lot. But I would like you to know that the mean fraction of body weight, blood, makes up about 8% of your body weight. I would like you to know pH 7.35 to 7.45, and we're going to talk about that a lot this semester. I would like you to know what I said a moment ago, that hematocrit is about 45% of whole blood. Now for lab, you will need to know the red blood cell count and the white blood cell count. And if you actually look in different textbooks, these numbers will be slightly different, and I'm not really concerned with you knowing exactly 4.6 to 6.2 million or 5,000 to 10,000, because if you look in a different textbook, they will be slightly different, but they'll always be in that ballpark. So I would like you to know roughly what the normal white blood cell count is, what the normal red blood cell count is. So that's been the overview of blood. Now I want to talk about plasma, and that's what we see here in these bags. If you've ever donated plasma, you've seen this. And plasma actually looks a little bit like urine. If you've ever seen plasma, you see it kind of looks like urine. And when we get to the kidneys towards the end of the semester, we're going to see that urine is actually very similar to plasma. So plasma has kind of this yellow straw-like color, and it's the liquid portion of blood. It makes up about 55% of whole blood. And here are some charts from page 673 of your textbook. Now, this is a lot of information, and again, you do not have to know all of this. Some of it I would like you to know. This is what do we find in plasma? and plasma is mostly water. Our body is mostly water. Plasma is also mostly water. Water makes up about 92% of the weight of plasma. Next, we have the stuff that is dissolved in that water. That's what will make it plasma, is all the stuff that's dissolved in it. And proteins are the big thing that we're going to talk the most about this semester. Proteins. Now, there's a lot of different proteins in plasma, and we're going to talk about them in practically every chapter. First, I want to talk about something called albumin. Albumin is the protein in our plasma that actually regulates fluid levels. And albumin is a solute. And remember from Bio 137, solutes pull liquid towards themselves. Albumin is a really big solute, so it pulls a lot of liquid towards itself. This is what keeps water in your blood so that it doesn't completely seep out into your tissues. Albumin, I like to say, when I talk about solutes, they pull liquid towards themselves. Solutes suck. They suck water towards themselves. So albumin, being a really big solute, sucks water towards itself. It keeps liquid, keeps water in your blood. Globulins. There's actually different globulins. There's alpha globulins, beta globulins, gamma globulins. So what are the differences? Alpha globulins and beta globulins. You've heard of these, you just don't realize it. When you hear people talking about HDL and LDL, when they're talking about cholesterol, globulins are related to those. Those are actually types of alpha and beta globulins. And beta globulins and alpha globulins are transport proteins. When something is needed to be carried around in your blood, but it's fat-based, or it just doesn't dissolve well in water, it has to be attached to something. Think about if you've ever taken oil and put it in water, what happens? It doesn't mix. So when we have something like cholesterol or fats in our blood, we don't want it to just float around undissolved in the water that makes up the plasma. We want to attach it to something to help it become soluble. And that's what those alpha globulins and beta globulins are. Now they transport stuff other than fat, other than cholesterol. And we'll see that later in the semester. But alpha and beta globulins, they're proteins, but they're specifically transport proteins. There's another type of globulin called a gamma globulin. And you've heard of these too. You just heard them called antibodies. Later in this chapter, we're going to talk about antibodies, but antibodies and gamma globulins are the same thing. Fibrinogen. Fibrinogen is a protein that's dissolved in our blood, but when we go to form a blood clot to keep us from bleeding, fibrinogen goes through some changes and it becomes something that is no longer soluble. So we'll talk about that later in this chapter also. Fibrinogen is a soluble protein involved with blood clotting. And then we see lots of nutrients that are in our blood. You do not need to know all of these. You don't need to know the values, absolutely not. But this is just to give you an idea of some of the stuff that we find in our blood plasma. Over here, electrolytes. Again, right now, you don't need to know all these numbers in every single electrolyte, but most of them you're familiar with because you talked about them a lot in Bio 137. Sodium and potassium, calcium and magnesium and chloride. We're going to talk about bicarbonate a lot this semester. This is something involved with maintaining our blood pH. Phosphate, sulfate. So you don't need to know all of these electrolytes, but you're going to kind of pick them up just because of repeated exposure this semester. Nitrogenous wastes. When our body metabolizes protein, so when you eat a steak and that steak starts to get broken down into proteins and then eventually into amino acids. One of the things that gets produced is waste that contains a lot of nitrogen. And here are the different types of nitrogenous waste that's produced by our body and carried in our blood. Some of them you've probably heard of, urea and uric acid. We've also probably heard creatine and ammonia. But these are just various types of nitrogenous waste. Again, you don't have to know all of these. You don't have to know the values. This is just to give you an idea of what's in your blood. And then other components. So dissolved carbon dioxide, CO2, dissolved oxygen, dissolved nitrogen, enzymes, hormones. All of this stuff is found in our blood, in our plasma. And here's a lot of what I was just talking about. This is also on page 673 of your textbook. It's just looking more in depth at those proteins, which I just talked about. Albumin, again, responsible for the osmotic pressure. Globulins, the alpha globulin, beta globulin, gamma globulin, transport, and antibodies. And then finally, fibrinogen. Now I want to talk about two words that we're going to use quite a bit in this chapter and in several other chapters this semester, and that's viscosity and osmolarity. And I want to group them under a bigger topic, concentration. Really, for this class, we can, anytime we hear viscosity or osmolarity, we can kind of substitute the word concentration, because for the depth that we go to in this class, they're kind of going to work the same. We can say concentration and it works just fine. But there is a little bit of difference between viscosity and osmolarity, and they play an important role when we talk about unloading nutrients to our body, or unloading liquid to our body, or when we get to the kidneys, how the kidneys cleanse our blood. So let's look at what the difference in viscosity and osmolarity are. So first, viscosity. By definition, viscosity is the resistance to flow. We're going to talk a lot about blood flow over the next several lectures. And blood flow, blood moves through our blood vessels. But if blood is more viscous, it means it has a harder time moving through our vessels. I like to use the example of if you pour water through a garden hose versus if you pour mayonnaise through a garden hose. Mayonnaise is more viscous. Which is going to pour through the garden hose easier, mayonnaise or water? Water, because it is less viscous. So viscosity is resistance to flow. The higher the viscosity, the thicker that liquid is and the harder it is for it to flow. We can think of viscosity as the thickness or the stickiness of a fluid. Your book uses mineral oil versus water, but most people aren't super familiar with mineral oil, but everyone has seen mayonnaise or ketchup or something like that. Blood is 4.5 to 5 times more viscous than water, and that's kind of regulated by the number of, or the amount of red blood cells. More red blood cells, the hematocrit goes up. When the hematocrit goes up, blood is thicker. It's more viscous. It has more resistance to flow. But if we just look at plasma, plasma is about 2 times as viscous as water, and that's because of the proteins dissolved in it. Now, keep blood flow in mind, because we're going to talk a lot about blood flow in the next 2 chapters after chapter 18 finishes. The other word we're going to see a lot this semester is osmolarity. And the book definition of osmolarity is the molarity of dissolved particles that cannot pass through vessel walls. That can be a little bit confusing. And another word we'll hear is the colloid osmotic pressure, or the COP. Colloid osmotic pressure is just the osmotic pressure of our plasma. So let's kind of unpack this and see what it's trying to tell us. If we just had plasma in our blood vessels, pretend that the red blood cells, the white blood cells, the platelets aren't there. Just pretend we have plasma, so water and all of the stuff dissolved in it is flowing through our blood vessels. Well, remember the proteins, especially that albumin, is pulling liquid towards itself. But that liquid is also trying to press its way out because of hydrostatic pressure. Hydrostatic pressure is the pressure of a liquid trying to press against the walls of its container, in this case blood vessels. And as that liquid tries to press its way through, the colloid osmotic pressure, the osmolarity, is pulling that liquid back in. So how do we figure out what the osmolarity is? The osmolarity is determined by the things in that liquid that are too big to pass through blood vessel walls. When we talk about capillaries later on, capillaries are really, really permeable. It's easy for liquid and really, really small things dissolved in that liquid to pass through them. But bigger things like albumin and things that are too big to get through those permeable capillaries pull that water towards themselves. When we measure that all out, the water trying to press its way out and the bigger particles trying to pull that water back in, that gives us the osmolarity. That'll make more sense in a couple of chapters when we talk about how things move in and out of the blood vessels at the capillary. But keep that in mind. When we talk about osmolarity, we're talking about things that are not able to pass through blood vessel walls, but instead pull liquid towards themselves. So this determines filtration and reabsorption, which we will talk about in chapters 20 and 23. Filtration, that's the stuff leaving the blood vessels at the capillaries. Reabsorption is that water that's getting pulled back in to the blood vessels at the capillaries. Now, when I play this, you're not going to be able to hear the audio. Unfortunately, it doesn't allow me to do that, but I'm going to talk about what this video is showing you, so I'm going to pause it off and on to talk about it. And you've actually probably seen a very similar video back in Bio 137. Now what we see here is water with some small particles dissolved in it. And there's a membrane dividing these two sides. And the membrane is going to be permeable to really, really small stuff. So like water molecules. But things like these big purple solutes are too big to pass. Now right now we can see that the concentration of particles in the water is the same on each side of that membrane. Hopefully this is sounding familiar because we did a lab about it in Bio 137. You saw an example of it in lab, you read a lot about it, but let's see what happens here. So what we're seeing here is, in this case, these particles, I guess, are small enough to pass. I think I just said that they're too big, but in this case, I guess they're small enough that they can pass. And it's going to show us here in just a moment something that's too big to pass and what effect that has. So things are moving across this membrane. Water and, I guess, these really, really small particles. Maybe this is oxygen or something like that. Let's roll that again. I accidentally hit the wrong button. So we're seeing things move back and forth. Now what we're seeing here, they say this yellow thing is something that is polar. So polar things don't like to cross membranes. The membrane repels them if they're polar. So we see they're not crossing the membrane in this case. Here's water. Now, water is polar, but it's really, really small molecule. So even though it's polar, it can still cross the membrane. Think back to Bio 137. Do you remember what it's called when water is diffusing across the membrane? It is a type of diffusion, but we give it a special name. It's called osmosis. So what we see when these water molecules are crossing the membrane, that's osmosis. But now we've got some really big molecules here. These are urea. It could be anything as long as it's really, really big. And we see that it cannot cross the membrane. And since it's polar, it's pulling the water towards itself. Water is polar. This urea is polar. So it's pulling water towards itself. In addition to that, it's a solute. What did we say? Solutes suck. Solutes pull liquid towards themselves. There's a greater concentration of urea on the right side than on the left. Typically, diffusion says everything wants to move from high to low concentration. So this urea wants to move the other direction, but it can't. It's too big. But it's a solute, so it's pulling water towards itself. Something else is going to happen. Let's see what happens. The water is going to get sucked towards these solutes because solutes suck. And look what's happening to the water level. So those things that are dissolved in the plasma that are too big to cross a membrane, they pull water towards themselves. And that's how our blood stays at the correct level of water. Now that's actually where I'm going to end this lecture. There's going to be another lecture where we talk about red blood cells and another lecture where we talk about white blood cells and maybe even another lecture where we talk about platelets. So I'm going to kind of break this chapter up into a few lectures, but you'll want to watch, several of them in one day so that you're staying on track with our course calendar. All right? Well, if you have any questions after reading and after watching this video, please let me know before you move on to the next video. I really want those questions and I want them asked when you have them so that you don't fall behind or get confused. All right? Take care.